So in this lecture, we're going to look at microbial genetics. Now, some of the genetics that happen in microbes are very similar to what happens in eukaryotes, including us. So some of this will look like a little bit of review from some of the things you've learned in genetics in A&P. However, and I'm like, there are some differences. One being the main example, and I'm like, is the fact that bacteria don't have a nucleus. So where some of this takes place is going to be different. But it's going to be kind of the basics, and I'm like the little central dogma of DNA to messenger RNA to a polypeptide, aka we're just making proteins using our genetics. So we're going to look at in this PowerPoint some basic terminology, structures, what's happening, how do we make more DNA, what are the basics of the central dogma, the transcription and translation, what's happening. Then we get into what happens if there's a mutation in the genetics, and as well as how can genes be transferred, how can we move and transfer genetic information. So starting with some basic terminology and structures. First, and I'm like, it's microbial genetics. So genetics, what is it? It's the study of inheritable traits as expressed in an organism's genetic material, which sounds like a mouthful. There are genes. We have DNA, which contains genes, which express various traits, and those are inheritable, which means they can be passed on. Now, a lot of times when we start talking about genes, we start hearing the word genome. What a genome is just the entire genetic makeup of an organism. All of its DNA, all of its genes, all of those nucleotide sequences. What are the A's, T's, C's, and G's? How many are there? What is the order of them? It's what are all the genes that code for various things. It's everything is an organism's genome. Now, a little review, because you've been learning DNA probably since middle school, and so some review about what do you know about DNA already, since we're going to be talking about DNA. And I'm like, and here are some things that may refresh your memory, but things you may already remember about DNA. 1, it's double-stranded. 2, it's a helix shape. 3, I'm like, A's pair with T's, C pairs with G's. There's a sugar phosphate backbone, and then the bases, those A's, T's, C's, and G's, the adenine, thymines, guanines, and cytosines, hang out in the middle, and they're attached together by a hydrogen bond. Now, and I'm like, are hydrogen bonds strong or weak? And I'm like, well, hydrogen bonds are weak, which most of the time you're like, well, why would we want weak bonds in our DNA? I'm like, well, the two strands are held together by weak bonds, which is good because when we need to make more DNA, we can pull them apart really easily and be able to put them back really easily. So kind of pull them apart so we can read it and then put it right back together. Also, a big key characteristic of DNA is the fact that it's anti-parallel, which means one strand goes one direction when the other strand goes the other direction. It's kind of like the street. One side of the road, they travel one direction, and the other side of the road, they travel the other direction. Now, one end of one of the strands we call the 5' end, the other strand we call the 3', which means the other strand over here goes the opposite. So the 5 is on this side, the 3 on this side. They're anti-parallel from each other, because that will come into play shortly, the fact that they are anti-parallel. Now, when we're talking about genomes, again, we're going to also talk about prokaryotic genomes, things that don't have a nucleus or nuclei. Now, when we're talking about their genomes, which is everything, all the genes, all the bases, we're talking about all the chromosomal DNA, any associated proteins that hold it all together, as well as some RNA. And I'm like, because there is some RNA in prokaryotes as well. Now, also, a prokaryote's genome, their chromosomes are haploid, which just means they only have one copy. All of our chromosomes, we pair up, and I'm like, we have 23 pairs. Chromosomes in prokaryotes, they're haploid, they're not paired. And I'm like, I always think haploid sounds like haploid. They're non-paired, it's half of a pair. They're circular versus linear and look like X's, and they're found in the nucleoid region because there is no actual nucleus. Also, prokaryotes may also have these small plasmids. And I'm like, which means they've got these small molecules of DNA. It's still DNA. This is its normal DNA that codes for everything that's needed in normal daily life. However, they also have these other little circular strands of DNA called plasmids. And I'm like, they do their own thing, they replicate on their own, they're not essential for normal metabolism, growth, reproduction, that's all of this DNA. However, they can have survival advantages because it's on these plasmids where you can find genes, because it's still DNA, and I'm like, that can code for resistance to things such as various antibiotics. And this is where our concern is, are these genes that code for resistance to antibiotics. Now, difference are eukaryotic genomes. We have at least one chromosome per cell. They like to hang out in pairs, so they're diploid, di meaning two. They're linear instead of circular, so they look like big lines, and they are found in the nucleus. Now, my little note about eukaryotes. Some fungi, which are eukaryotes, and some protozoa, which are eukaryotes, can carry plasmids as well. They can carry those little bits of DNA. I'm like, but we don't. But this is showing you how we can fit, because this little purple part, this is our DNA. And we can fit 6 feet of DNA in one single nucleus of one of your cells. That's really, really skinny, but it wraps around proteins and these nucleosome structures, and then we wrap all of those around. Eventually it starts to look like the chromatin, and it condenses, and we start to see things called chromosomes. But we've got really long, really skinny DNA inside the nucleus of each of our cells. Now, we also, when we're starting to replicate cells, when they reproduce, obviously you need 2 of everything now, and that includes replicating DNA. We need double the amount of information, double amount of DNA. And so for DNA replication, we are building something. We're making more when we replicate. And so it's an anabolic process. Now, the 2 main things we need for any type of anabolic process: we need monomers, are our building blocks, and we need energy to put them together. What's nice in organisms, we have what are called as triphosphate deoxyribonucleotides, which is a really massive long type of word. But what's nice is that this one molecule serves both functions. It has the monomer and it has the energy. And so it has the bases. It's DNA. It has the bases. This is a guanine base. However, attached to it, we have this high energy bond, triphosphates. If you think of the last time you've also heard of triphosphates, it's your ATP, adenosine triphosphate. Those are high energy bonds. Well, now we put these high energy bonds attached to DNA, and we have the energy, and we have the building blocks, and so when we start putting this together, we can release the energy by removing some of these phosphates. And so we've got this awesome building block to do all of this. Now, also when we make new DNA, the way we do it is we have the original strand, which is double-stranded. We will kind of tear it apart. We'll put it back together, but I'm like, we pull it apart, and each strand becomes a template for one of the new strands. And so every time we make new DNA, you're going to have an original strand, and you're going to have a new strand. This is called the semi-conservative model. And again, we didn't know for a long time that that's how we did this. We weren't sure if we just took one double strand, it stayed as a double strand, and we just made a whole completely different double strand that was all brand new. Well, now we realize that all of that double DNA, all of that new DNA, one is an original strand, one is a daughter strand, or a copy. So it's a semi-conservative. So you can see, here's the original DNA, we pull it apart. In your new DNA, one strand is the original, one strand is that copy. Now, when we want to make more DNA, it's not just simple, here, let's just make more DNA. And I'm like, there's kind of a 3-step process that happens in DNA. Yes, I love Big Bang. Here's my somewhat inappropriate Big Bang meme, but it totally works for this. Kind of the main part. DNA is held together by hydrogen bonds. Those bases that are inside of here held together by those weak hydrogen bonds. Well, there's an enzyme called helicase that unzips the DNA. It pulls apart those hydrogen bonds. Hence, this is the helicase. Now, as we unzip it, it travels down the DNA, pulling it apart, unzipping or separating those hydrogen bonds. we have to also add these stabilizing proteins. Main reason, we are going from a helical shape to a straight shape. And if you've ever tried to take like an old telephone cord and try to straighten it out, it doesn't want to go straight. It wants to stay in a helical shape. And so we have these stabilizing proteins to try to prevent it from winding up on itself as we're trying to make new copies of it. Then we have another enzyme, so again they're all ending in an ACE. DNA helicase unzips, stabilizing proteins keeps it from winding back up, and then we have DNA polymerase that's going to bring in new bases, bring in new A's, new T's, new C's, new G's, and it will always add it to the 3' end that it is making. So if this is an original strand, your new strand will be here, and if this is the 5' side, and I'm like, here, this will be the 3' on this one. And so always add on this side. And I'm like, which this can become a problem when we have strands going 2 different directions. And so here again is showing 2 different strands. One is going to show you what's happening on what we call the leading strand and on the lagging strand. And so here's my strand, here's a 3' side, that's the original. And then my new daughter strand, or my copy, again, it's going to go anti-parallel, so here's the 5' side, here's the 3' side down here. And DNA polymerase always adds these bases onto the 3' side. So it's going to bring it in and just keep adding more and more bases, matching up A's, T's, C's, and G's. Now, it's called the leading strand because this is not an issue. As we unzip the DNA and make more copies, we can continually add more bases onto this new 3' side. Our problem becomes down here with this other strand, because if this is the 5' side, this is the 3' side, and we have to be adding on this side. But we're traveling this direction, but we have to add onto this side, and so we're almost having to add backwards, which is not the direction we want to be going. So we call it a lagging strand. So what ends up happening is we have to lay down these little primers, and it does go in reverse. And it'll, find it where there's another primer, and I'm like, and we have these little segments, they're called Okazaki fragments. And I'm like, that we lay down a primer, go in reverse, make these little snippets of DNA. Lay down another primer, go in reverse. It's bringing in the bases, but going in opposite directions. We'll remove these primers, and I'm like, and fill in the gap so it all is one continuous DNA molecule. But I'm like, we just have this constant going in reverse. So it takes a little bit longer. You can't just unzip and continually go along. We have to keep adding and going backwards and adding and going backwards and then putting them all together. An enzyme called ligase is an enzyme that really kind of binds all of these little tiny strands together. Now, I do have a video, and I'm like, that shows this happening too. And I'm like, the leading strand versus the lagging strand. Let's see if I've got audio. Where's my audio? Here we go.
By the enzyme helicase.
When DNA replicates, its strands are separated by the enzyme helicase.
Single-stranded DNA binding proteins keep the strands from re-annealing.
Single-stranded DNA binding proteins keep the
One
strands
DNA
from
strand
re-annealing.
encodes the leading strand, which forms from its 5' to
One
its 3'
DNA strand
end,
encodes the
using
leading
DNA
strand,
polymerase
which
3.
forms from its 5' to its 3'
No problem
end,
here,
using
but
DNA
the lagging
polymerase
strand
3.
presents problems. It has to
No
form
problem
from 5'
here,
to
but
3'
the lagging
too.
strand presents problems.
It forms in
It
pieces
has
called
to form
Okazaki
from 5'
fragments.
to 3' too. It forms in pieces called Okazaki fragments.
First, an RNA primase lays down an RNA primer. Then,
First,
DNA
an
polymerase
RNA
3
primase
lays down
an
new
RNA
DNA.
primer. Then, DNA polymerase 3 lays down new DNA.
The process repeats again and again.
The process repeats again and again.
DNA polymerase 1 replaces the RNA primers with DNA.
DNA
Finally,
polymerase 1
DNA ligase
replaces the
links the
RNA
Okazaki
primers
fragment.
with DNA. Finally, DNA ligase links the Okazaki
And so
fragment.
you've got it all together. I'm like, but yes, those, that lagging strand just presents a problem. It's not a bad problem, it just takes sometimes a little bit longer. Now, away from making new DNA, we can make more DNA. Now we're going to talk about the difference between a genotype and a phenotype. a genotype are all the genes that you have in your genome. And I'm like, so a gene codes for something. You have a gene that codes for your eye color. We actually have several genes that code for eye color. And I'm like, you have genes that code for skin color and hair color and any other characteristic about you. You've got a gene that codes for it. And I'm like, all of your genes includes your genotype. Your phenotype is what you actually look like. These are physical features or various traits that you can do. And I'm like, based on your genotypes. Your genotype are all your genes, but the phenotype is what they code for. So the genotype, you've got a DNA segment that codes for hair color, several of genes that code for hair color, and your phenotype is what color is your hair. I'm like, but it doesn't always have to be a physical feature. It could be functional traits, whether you have genes that code for breaking down milk and lactose, or whether you don't have genes that code for breaking down lactose and you're lactose intolerant. And I'm like, so you have genes are functional traits as well. Now, my little note on why this is not an equal, because normally your genotype codes for a phenotype. They should be equal. And I'm like, however, there are some genes that get turned off, which means they are not expressed. This even happens in the gene that codes for lactase that breaks down lactose. And I'm like, at some point in your life, you may have been able to break down lactose, but then some other time in your life, you can't, because the gene starts to get turned off. And I'm like, or the fact your hair color changes, or eye color changes, or skin color changes, or, anything else that changes about you. A lot of times that's genes that are getting turned on and off. So it's not always the gene is going to be turned on coding for the phenotype, but they do code for phenotypes. Now, transfer of genetic information. So the DNA holds all the information we need for doing anything in our body. And there are 2 steps, and I'm like, that we can use to take that information in our DNA and create other parts of our cell. Now, transcription is taking DNA and turn it into RNA, specifically messenger RNA. And then translation is taking RNA and turn it into polypeptides. A polypeptide just means a long chain of amino acids. We're going to create proteins, because proteins are chains of amino acids that get folded just right. And we have proteins. They are needed for growth, they are needed for metabolism, they're needed for regulation, they are needed for structural materials, they're needed for enzymes. We need proteins to do just about everything. So now we've got our, 2-step process, taking that information in DNA to eventually make any protein we want. So we call it the central dogma of genetics. How do we take this double-stranded DNA? How do we make our messenger RNA? And then how do we take that message on the messenger RNA and code it into a language of amino acids? Transcription just means to make a copy. Translation means to change it into a different language. So we're going to change this from the language of RNA to the language of proteins, which are amino acids. And it's these chains of amino acids are proteins that'll code for some type of phenotype. So, some review or some little information on transcription. One, again, all the information in DNA is copied as RNA. Kind of my example with transcription, the word transcribe means to make a copy. If you're a transcriptionist, you work in the courthouse and you just make copies of everything. You just write down what you hear. You're making a copy. And I'm like, it's not the exact same, because one came from a person's mouth and the other one is typed up, but it still has all the exact same information. That's what we're doing with DNA to RNA. And I'm like, it's not the exact same because it's RNA, not DNA, but still contains all the information that's needed. Now, to go from DNA to RNA, we need an RNA polymerase. It's it's an enzyme, the L-indinase, almost. And I'm like, that makes RNA. And then we need our RNA nucleotides. A's use G's and C's. We lose the T, and it becomes the U. Now, other things that can help, an RNA primer is usually laid down. We have a ribosomal RNA, a transfer RNA, and a regulatory RNA. And I'm like, that all can help. I'm not going to concentrate on those, though. Now, I was going to not show it you yet. And I'm like, let's see if you can try transcription. And I'm like, of what binds, if the T is on your DNA, we're only copying one strand of DNA. If the T is on your DNA, what would the RNA nucleotide be brought in? Be an A. And if we have an A on our DNA, would bring in a U, and a C to a G, A brings in a U, T pairs up with A, G pairs up with C, and so on. There we go. And I'm like, so it constantly is binding. Still, C's are always with G's. And I'm like, if it had a T, it still pairs up with an A. But if it has an A, we don't have T's. We have U's instead. So I'm like, same basic idea. That's your transcription happening. Now, I have another little video clip of this too. See if I can make it any louder.
The innermost workings of how a simple code is turned into...
There we go.
The innermost workings of how a simple code is turned into flesh and blood. The innermost workings. This is what Francis Crick called the central dogma of modern biology. How DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography,
Now, my little note, because we're going to end right there and pick it up. That's right where transcription ends, is we took our double-stranded DNA, we unzipped it, and we made our single-stranded messenger RNA. And that does not matter if that happens on a prokaryote or a eukaryote. Same thing, same basic idea is still going to happen. But now we need to take that messenger RNA that we just got done making and translate it into that language of amino acids to make our proteins. And so our participants, we've got our messenger RNA, transfer RNA. Now the transfer is called transfer for a reason because it transfers or brings in the amino acids. And then we have ribosomes, because if you remember, you've been drilled into your head, ribosomes make proteins. But this is how, because it's where translation takes place, and we're making proteins in the ribosomes. And ribosomes, on ribosomes, they also have another type of RNA found in their structure called ribosomal RNA. So we've got our messenger RNA template, transfer RNA is going to bring in the amino acids, and ribosomes are going to be there because that's where everything takes place. Now, this is showing what a transfer RNA looks like. They kind of look like weird little shaped T's a little bit. Humor me on that. And I'm like, and the reason why it knows how to bring in the correct amino acid is that on one end of the transfer RNA molecule, it has a 3-letter code that pairs up with the 3 letters on messenger RNA. It reads messenger RNA 3 letters at a time. Every 3 letters, a messenger RNA is called a codon. So every 3 letters, AUG is one codon, and it codes for this amino acid. CGG is another 3 letters, it's another codon, and it codes for this amino acid. Well, this particular transfer RNA has an anticodon. It pairs up with the opposite of this, because it pairs up with the opposite code. And the opposite of ACC is UGG. So if there is a UGG, it's not on this one, but if I was reading backwards, and I'm like, but if there was a UGG, this transfer RNA would be like, that's my that's me, I'm up, that's my match. And I'm like, for my anticodon on me, and it knows then which amino acid to bring in. Now, and I'm like, as it reads 3 letters, some review too, there are 20 different amino acids, but there are 64 different ways you can mix around A, U, C's, and G's into groups of 3, into codons. And I'm like, so there are 64 different ways here, but yet only 20 amino acids. And I'm like, so if you'll notice, there are several codons, several 3-letter sequences that all code for the same amino acid. That's fine. Also, if you notice, there's one 3-letter sequence. It codes for an amino acid called methionine. We usually shorten it to 3-letter abbreviations. But this is always going to be the start codon. Transfer RNA is always going to be looking for an AUG to bring in this first amino acid and start everything off. And so these are the first 3 letters that's going to bring methionine in, and now we can continue to read the rest of the messenger RNA. And it will continue to read as much messenger RNA that's there, or until it reaches one of these 3 different stop codons. Now, there's called stop codons because there's not an amino acid that codes that matches up with any of these. There are a couple prokaryotic organisms, though, that do have an amino acid for UGA, rare. In eukaryotes, there's nothing. And I'm like, but it's kind of our unique one. And I'm like, there's also this other random amino acid that some prokaryotes too have too. Not worrying about them. And I'm like, these are 3 stop codons that it will continue to read. And just every 3 letters, bring an amino acid till it hits one of these stop codons. So we've got these different letters, and I'm like, in your book, it's page 205. And I'm like, because this page 205 will help when we get to the in-class activity. And I'm like, but that's where we find this particular table. Now, how to use the table, it's a 3-letter sequence. For example, AUG, because we're going to do my little code here. My first 3 letters are AUG. Now, how we use the table, the first nucleotide base is the A. So we've got UCAG. First letter is an A, so we're nowhere in this row. The second letter was U, and our second base is up here. And so we've got UCA or G. Well, for us, it's the U, AU. And then our third letter of our codon is G. Like, we know we're in this row, we know we're in this column. Way over here, we've got the 4 choices for that third letter. And our third letter is G. And so we know we're in this last row for AUG. So I'm like, that's where we get, and I'm like, AUG codes for methionine. Now, let's see if you can find UAC on this table. Again, UAC. First letter is U, so you have to find the correct row for U's. And then the second letter was A. And I'm like, so you have to find the A across the top for our second base, so we know what row and what column we're in. And then the third letter was C. And over here it tells us where that third letter's position is. And here we have UAC, of tyrosine. So I'm like, we've got the rest of the codes. If you want, you can pause me right now, find page 205, and see if you can fill it in. And then, you can pause me. So I'm giving you a second if you wanted to pause me. And I'm like, because otherwise I'm going to show you the answers. And I'm like, of what that amino acid sequence, that polypeptide, which is just a chain of amino acids, is going to be. And here it is. And I'm like, it's your start or methionine, tyrosine, glycine, phenylalanine, and then we run into a stop codon, UAA. And we stop. And I'm like, we don't read it any farther. Here's our amino acid sequence. Amino acid sequences are usually hundreds, if not thousands, of amino acids long. Mine was 4. And I'm like, for my example. Also, there is a little bit difference in ribosome structure between a prokaryote and a eukaryote. Same basic idea. They both are helping read the messenger RNA and make proteins, but a prokaryotic ribosome is a little bit smaller. They call it as a 7ds size versus an 80s. Either way, a ribosome is made up of 2 units, and those 2 units, it's like if I take my 2 hands and clap them together, it literally chomps on to that messenger RNA. So the 2 parts of the ribosome, whether it's prokaryotic or eukaryotic, will chomp on to the messenger RNA and start reading it. Now when it does do translation, it does do it in kind of a 3-step process. And I'm like, the first is initiation. It's finding a start codon. It's finding that AUG. That's where the ribosome is going to chomp on and start binding. So you can see the 2 parts recognizing that AUG, they're coming together. And then we're going to have the elongation phase. The elongation is where these transfer RNAs, they are reading 3 letters at a time, bringing in the appropriate amino acid that codes for them, and then dropping it off. And you can see off the top, these each are the new amino acids that we're stringing together. As we are traveling along our messenger RNA, we are bringing more and more amino acids together and attaching them. And then we will continue to do this till you hit one of those 3 stop codons. And then we have our termination. And then whatever is there, whatever that amino acid sequence is that we just made, it will get released, it will get folded and turned into its appropriate protein. So there are kind of 3 sites that are happening, based on your initiation, elongation, and termination all happening here. So inside the ribosome, we have the A site. This is where the reading is happening. The P site is where you're attaching the amino acids together, and the E is where that messenger RNA is exiting. So it's E for exit. And I'm like, so you can see approaching, exiting, polypeptide or peptide bonds are forming, something like that. It spells ape backwards. I mean, it's forwards because this is the direction the messenger RNA has come in. Now, one thing that whether it's us or prokaryotes can do is reading messenger RNA can take a long time. And so we have to have proteins everywhere, thousands of them, millions of them in our body. Every second we have to be churning out proteins. To speed this up, we can have multiple ribosomes all reading one messenger RNA. And I'm like creating lots of copies of proteins and reading that messenger RNA a whole lot faster. Here's an actual picture of that happening. And so I'm like, it's a little bit faster. And whether it's a prokaryote or a eukaryote, both can do that. Now, we're going to finish watching that video. So that first part of the video showed how we took apart the DNA, made our single-stranded messenger RNA, and it left the nucleus. Granted, in a prokaryote, they don't have a nucleus. It just all happens in the nucleoid region in the cytoplasm. And I'm like, but we're going to see what happens to that messenger RNA, how it gets read by the ribosome. And you'll see the 2 parts of the ribosome kind of clamping on together and starting to read it and bring in the appropriate amino acids.
The innermost
Let me skip to where we left off.
Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
So yeah, we gotta make a lot of protein and really fast, and that's just one protein that we need in our body.
Our bone marrow
Now,
churn out
comparing
100
prokaryote
trillion
versus
molecules of
eukaryote,
it per second.
I'm like, the difference is, same basic ideas are happening in the central dogma. And I'm like, differences though. One, transcription is going to take place in the nucleus in a eukaryote, because there is a nucleus, and that's the only place you'll find DNA is in the nucleus. And I'm like, in a prokaryote where there is no nucleus, this is all going to happen in the cytoplasm. Also, a difference again is just looking at the structure of DNA. And I'm like, in a eukaryote, our DNA is linear. It's kind of folded here, but it's linear. And a prokaryote, their DNA is circular. So it just has to find on that circle, where is that start codon for it to start. So kind of just where transcription takes place is a little bit different. Otherwise, same basic ideas that are happening. We make that messenger RNA. The ribosome is going to find it, chomp down on it, start reading it, and it can do it in several places. A nascent protein just means it's a building or a growing protein. So we're making those proteins there as we read the messenger RNA. So just a little review. Yes, the central dogma of genetics, and MIC is taking that DNA, making it into messenger RNA, and then translating it into the language of proteins, which is amino acids. And I'm like, and we read, page 205, and I'm like, we can read these 3 letters at a time, bringing in the correct amino acid. Yay! Now, because I want to get through mutations on this, what happens if we screw it up? And I'm like, because it happens. And I'm like, they're called mutations. It just means we have a change in nucleotide base sequence. Means somewhere we didn't do DNA replication correctly, or we didn't do transcription correctly, and it's not good. Now, mutations themselves are rare, and I'm like, and when they do happen, there are times that, at least in our bodies, I'm like, we can fix it. And a lot of times we've got ways to go back and correct those errors. If not, it usually kills the cell. Usually, mutations generally are not good. On rare occasions, and I'm like, a mutation can lead to an improvement. There are times that I was going to say, here's my random tiger bunny, and I'm like, there might be a mutation that makes a bunny have the tiger stripes. Well, if it's trying to blend into tall grasses, that would be an advantage. And I'm like, a random mutation all of a sudden turned into some type of advantage that allowed it to survive and pass on those traits. That's called natural selection. If you all of a sudden have some trait that allows you to survive and pass on that trait, natural selection. And I'm like, but that's rare when we start to have these beneficial mutations. Now, we do group mutations, and I'm like, on gross mutations, meaning a huge change in DNA, versus point mutations, which means one single base changes. Now, having one base change doesn't mean, oh, well, that's not that bad. It can still be really bad, because there are different types of point mutations. And you're going to have to know these for your in-class activity. So here's my normal DNA. Normal DNA, it codes for my normal messenger RNA sequence, and then I read 3 bases at a time, and here's my normal amino acids that would form. Now, we've got several types, because there's two slides here, of point mutations. So one base change. And so in this case, my original base was an A on DNA. Change it to a G. So now a C gets brought in. However, this is called a silent mutation. The nucleotide changed, but there's no change in amino acid, because whether it was UUU or UUC, both of those codons code for the same amino acid of phenylalanine. So it's called a silent mutation. The DNA changed, but the protein didn't. We didn't change anything about the protein. So if you have to have a mutation, that would be the best one. Now, a missense mutation means you change one base. So this was originally a G up here. Changed it to a C, which then a G got brought in instead of the C. And I'm like, a missense mutation just means one amino acid is changed and that's it. So phenylalanine is the same, tyrosine, arginine, these are all the same. It's just this one amino acid is different. And then people are like, well, that's not so bad then either. You know, it really depends on which amino acid and which gene is being changed. And I'm like, one single amino acid among a chain of 1,000 can still make serious changes to the body and even cause death. And I'm like, example of a missense mutation that still causes us serious side effects by just one single base and one amino acid change is sickle cell anemia. And I'm like, there is, yes, and I'm like, a genetic code that codes for that normal red blood cell shape. And I'm like, however, one single base change, which codes just one amino acid change, every other amino acid, there's about 1,000 amino acids, by having just one amino acid change, and I'm like, now you have irregular shaped red blood cells and you have sickle cell anemia. So, and I'm like, you're like, it's not so bad. It is really not. And I'm like, you know, when you're talking how many could be changed, but it could still have really serious side effects. Another type of mutation, here's my normal up here again, is called a nonsense mutation. It just means the change that I made in my DNA sequence coded for a stop codon. That's not good, because now you're never going to finish making your appropriate protein. And I'm like, and you just can't anymore. Usually leads to death. And then the last are called frame shift mutations. It just means we add one base, in this case we adding one T, or we remove a base. We removed a T. And I'm like, because originally it starts out as 4 A's and a T. So here we added an extra T in there, because here's my 4 A's and a T. And I'm like, we removed a T. Now, why frame shift insertions or deletions are so bad? Even though we're only adding one base in, it literally shifts the entire genetic sequence down. We have to read 3 bases at a time. But if I add one base in, all of my codons are going to be 1 letter off, which means every single amino acid after that insertion or after that deletion are different from the original. And so frame shift insertions or deletions, just with that one base change, can be extremely detrimental. Now, ways that mutations can happen. One, we call them spontaneous mutations. It just means there's some type of error somewhere in DNA replication or repair. We're constantly making new DNA, we're constantly making RNA, and sometimes when we're making all this new stuff or repairing any of it, we can accidentally bring in a wrong base. It's called spontaneous, just an accident happened. And I'm like, more common, and I'm like, are mutagens. Mutagen just means they increase the likelihood of a mutation from happening. The word means generating a mutation. They increase, they increase their likelihood. Now, we've got different types of radiation, ionizing and non-ionizing types of radiation. Both are mutagens. Examples of ionizing radiation are X-rays. And I'm like, there's a reason why when people have X-rays taken, the X-ray tech like runs out of the room, or you put the big lead vest on. I'm like, because it increases the chance of a mutation in your DNA from, to happen. Non-ionizing radiation, UV light. There's a reason why they tell you not to go in the sun all the time, because UV light increases the chances of mutations happening, which mutations can lead to cancer. And I'm like, it can start to cause, and I'm like, your cells to start to reproduce. And then we have chemical mutagens. One, we have nucleotide analogs, which just means, again, they look like the nucleotides. They look like adenosine, thymine, cytosine, guanine. And I'm like, they look like them, but they're not, which means wrong bases get brought in. And then we also have nucleotide altering chemicals, which just means they alter the structure of the nucleotide. So your adenine, cytosine, guanine, thymine, they don't look normal, which means they're not going to perform normal. A big example of an organism that can do that, and I'm like, is the fungus Aspergillus actually produces a toxin that can change the nucleotide shape. And yes, it causes mutations that can have serious side effects. Now, when we talk about mutations, and I'm like, 2 terms that are used, mutants and wild types. Now, I'm going to start with the wild type, and I'm like, a wild, all the people are like, it's wild, that's weird, whatever. Wild type, and I'm like, just means these are cells and with normal DNA, nothing's changed that are normally found out in nature. And I'm like, that's called a wild type organism. And I'm like, a mutant means, and I'm like, there was at some point some type of mutation. It never got repaired, and it started to reproduce. And all of the offspring, all of the descendants of that mutated cell are called mutants. They have that mutation. And I'm like, so if your dad had a mutation or your mom had a mutation and they passed on that mutation to me, I hate to say it, you are a mutant. And I'm like, they passed on a mutation to you. And so we talk about mutants, they have a mutation, wild types, normal types. Now, luckily, I'm like, mutations are rare. And I'm like, they don't happen all the time, or organisms would just be dying all the time. And I'm like, however, different types of mutagens increase the chance of having mutations happen by a rate of 10 to 1,000 times. Again, wearing the sunscreen, and I'm like, keeping out of UV light or different, not exposing yourself to different types of radiation. There are different type of chemicals that are mutagens. They're called carcinogenic. They can cause cancer because they can change DNA, the DNA somehow. And so I'm like, it can happen, and I'm like, and it's generally not good. Now, this is where we're going to end on here. I have a little bit left on the PowerPoint, but we're going to talk about that in lecture, and that's how we can transfer genetic information, how one organism can take its genetic information and pass it on or give it to another organism. So it's kind of the, how can we start to switch things around? How can I give my genetic information to another organism's genetic information?